Hi, my name is Vanessa Ward, and I'm here to discuss the big deal about obesogens, the link between endocrine disruption and obesity. As you may have gathered, the purpose for the following presentation is to examine the possible link between endocrine disruption and obesity. I will begin by providing a brief background on information pertaining to cell signaling, the endocrine system, and endocrine disruption. Afterward, I will provide some introductory evidence of a link between endocrine disruption and obesity, followed by the nature of exposure to endocrine disrupting chemicals and the biological basis for the mechanism of action. I will conclude with a risk assessment and my feelings of the actual threat we are facing. The last thing I will cover will be recommendations addressing this threat and possible actions that can be taken to address the issue concerning endocrine disruption and the role it is playing in the epidemic rates of obesity. In multicellular organisms, there's a complex network of cell signaling that coordinates growth, differentiation, and metabolism. Cells have a way of talking to each other through signaling molecules that are synthesized by signaling cells and produce a response in target cells by binding to a specific receptor protein. One system involved in cell communication is the endocrine system. The endocrine system is a system of glands that releases hormones that influence almost every cell, organ, and function of our bodies. Hormones that control the action of transcription factors can either promote or inhibit the expression of a gene. For this reason, the establishment of these interactions among cells is vital to the development of many tissues. Before cells become specialized or differentiated, they have potential to become any type of cell. Hormones serve as a way to tell these cells what type they are destined to be. And it is a delicate process. And in the same way your tone of voice can alter the meaning of a sentence, a slight change in the hormonal conditions can change the message telling the cell what it will be. We are faced with a problem referred to as endocrine disruption. This is when molecules similar in chemical structure to our naturally occurring hormones are recognized by receptors as actual hormones rather than other compounds. Both naturally occurring and synthetic chemicals can mimic hormones and result in abnormal activation or inhibition of gene expression. Today, obesity is occurring at epidemic proportions. In fact, 30% of adults were classified as obese in the United States in 2002, and today, 1 in 6 adolescents is obese. These shocking statistics makes one wonder if there's more to blame than just poor diet and lack of exercise. And this is where the theory of endocrine disruption and obesity comes in. Earlier, I talked about cell differentiation, and now I want to address one type of cells in particular, and those are adipocytes, or fat cells. Adipose cells form between the 14th and 16th weeks after fertilization. After the 23rd week, the total number of fat lobules remains fairly constant, but from the 23rd to the 29th week, there's an increase in size of the lobules. This suggests that the 14th to the 23rd week is a sensitive period, particularly to adipocyte formation, and that disturbances of normal adipogenesis during this time may play a role in the risk of obesity later on. Originally, there was no connection being made between chemical exposure and obesity, since the main focus was on effects of endocrine disruption on reproduction and behavior. Even though it was later determined that the test animals were heavier than unexposed subjects, scientists who had been working with the mice for so long simply overlooked this trend. After some time, though, a pattern was detected, and scientist Retha Newbold, developmental biologist in the toxicology branch at the National Institute of Environmental Health Sciences, during one interview stated that I can tell you simply by the size of the animal which is a DES-exposed mouse and which isn't. In 2006, after some initial observations, there did seem to be a pattern in increased rate of obesity and the increased use of chemicals that are known endocrine disruptors over the past 40 years. It was Bruce Bloomberg who came up with the term obesogens, which have been defined as a class of chemical compounds foreign to the body that are hypothesized to disrupt normal adipose cell development or homeostasis of metabolism of lipids, ultimately resulting in obesity. It's not known whether obesogens have the same effects on adults, but Bloomberg believes that the damage has been done and the effects will be clear on Americans born after World War II, when exposure to industrial chemicals increased. Some chemicals that have been hypothesized to interfere with adipogenesis are organotins, bisphenol A, and diethylstilbestrol. Organotins are compounds used in paint for ships, plastics, and fungicides, such as tributylin, or TBT. Bisphenol A and diethylstilbestrol, or DES, are chemicals similar in structure to estrogen. Bisphenol A is found in plastics, and DES was prescribed in the past to prevent miscarriage. Some sources of exposure to these chemicals include contact with fungicide-treated areas, contaminated food sources, especially seafood, plastic containers, and paint used for ships or boats. Although humans are at the greatest risk, exposure to these chemicals is definitely not limited to humans, but poses a risk to a wide range of species in contaminated areas or those that eat fish. Obesogens are involved in the alteration of adipogenesis. The retinoid X receptor, Rx-alpha, Rxr-beta, Rxr-gamma, and the peroxisome proliferated activated receptor, or PPAR-gamma, are both nuclear receptors that are involved in lipid homeostasis and adipogenesis. It has been proposed that chemicals that act on these receptors at times they are not normally acted on could lead to the formation of more adipocytes and possibly obesity. This diagram shows the signal transduction pathway of the Rxr and PPAR-gamma receptors and how tributylin and organotin could possibly modulate adipocyte differentiation. On the right side of the diagram, the black arrow labeled adipogenic gene indicates that the binding of TBT initiates transcription of a factor that promotes terminal differentiation of adipocytes. Since this area of research is so new, data is limited, but several studies have been conducted to support the hypothesis. For example, an in vitro study using a 3T3L1 cells, which are mouse embryonic fibroblasts similar to adipose cells, were treated with tributylin. Tributylin acted as a receptor agonist, meaning that it caused a response, and in this case, the response was the formation of more fat cells. TBT resulted in the expression of adipocyte-specific genes at a higher frequency. The relevance of this means that increased expression of adipocyte-specific genes would result in formation of more fat cells. In vivo studies have also been performed. In a study that exposed mice to DES in utero, it was found that doses of 10 to 100 micrograms per kilogram resulted in decreased body weight for life, and doses at 1 microgram per kilogram resulted in increased body weight as adults, along with the increase of percent body fat. Here we see the weights of mice following neonatal DES exposure. Although the DES-treated mice were born at a weight significantly lower than controls, by age 1 month, the DES-treated mice caught up with the control group, and by 4 months were much heavier than the controls. It's strange to think that two groups of mice who only differ in their exposure to DES could show such differences in weight, and this implication definitely has relevance to the increased rate in obesity and endocrine disruption.